So the ZUMA-1 study is the first multi-center trial of CD19 CAR T-cell therapy with axicabtagene, ciloleucel in patients with refractory aggressive B-cell non-Hodgkin lymphoma. So this study was started off as a phase 1 study where we treated 7 patients. And then after establishing the safety and feasibility, we went on to do the pivotal phase 2 portion of the trial where we treated a total of 101 patients. We enrolled patients with 3 different subtypes of lymphoma, and that included patients with diffuse large B-cell lymphoma, primary mediastinal B-cell lymphoma, and transformed follicular lymphoma. So these patients, to be eligible for the trial, they had to have refractory disease to the most recent line of chemotherapy, or they should have relapsed within 12 months after an autologous stem cell transplantation. And the primary endpoint for the pivotal portion of the study is the overall response rate.
So what did you find?
So what we are presenting at this meeting is the combined analysis of the phase 1 and phase 2 for a total of 108 patients, and we have a median follow-up of 15.4 months now. And what we observed at this time is that 42% of these patients still remain in remission at 15.4 months, and 40% of them remain in complete remission. The median duration of response for the complete remission patients has not been reached, and we have also not reached the median survival. In fact, at the 1-year time point, 56% of these patients are still alive. And at 18 months, we project that 52% of them are still going to be alive.
What about adverse effects?
So most of the adverse effects that we observed on this trial were observed within the first 4 to 8 weeks after the infusion of the axicabtagene product, because this is a single infusion. And the common adverse effects are cytokine release syndrome and neurological toxicity. The cytokine release syndrome was seen in majority of the patients, but most of them were grade 1 and grade 2 adverse events. And we observed grade 3 or higher cytokine release syndrome in about 13% of the patients. In the second most common toxicity we observed is neurological toxicity. And this has been, most of the neurological toxicities that we observed were grade 1 and grade 2. In about 28% of the patients, we found grade 3 or higher neurological toxicity. We now have long-term safety data as well on this study. Beyond the 6 months, we didn't find any new onset cytokine release syndrome or neurological toxicity, and we did not see any axicabtagene-related deaths after the 6-month time point.
So what is the conclusions, the take-home message from this research?
So this study now with the longer follow-up establishes that axicabtagene is a highly effective form of therapy. In fact, we found that 82% of these patients respond with a single infusion of this treatment, and 58% go into complete remission. At 15.4 months, 42% of the patients are still in remission, and the median survival has not been reached. So this data is consistent with what we've observed previously on single institution phase 1 and phase 2 trials, where about 40 to 45% of the patients appear to achieve long-term durability lasting many years. And it's likely that these patients may be cured, at least a significant proportion of these patients may be cured, but longer follow-up is needed. And this study also now confirms that long-term safety of this product. We've also found that we've done some preliminary analysis to understand the mechanisms of resistance with this product. We've analyzed tumor biopsy samples at the time of progression, and we found that in about a third of these patients, the actual target protein, the CD19 protein, is lost, which suggests that in order to further improve the efficacy of this product, one way to do that is to target multiple molecules on the surface of the tumor besides CD19. We've also found that in these biopsies that were obtained at progression, another molecule called PD-L1 appears to be upregulated on these tumor cells. And PD-L1 is an inhibitory ligand which can suppress the function of CAR T cells. So another way to improve the efficacy of this product is to do combination studies where we combine the CAR T cell therapy with a PD-1 or PD-L1 antibody, and those studies are ongoing at this point.
When will they be reporting any data?
So at this meeting, we are reporting the data from the first 6 patients who have been treated with a combination of CD19 CAR T cell therapy with PD-L1 antibody, and that's being presented tomorrow.